Assalamualaikum everyone. Welcome to the first biology class of the A levels, and we are going to look at the first chapter, which is the cell structure. So I think this might be the easiest chapter out of all the chapters that you have in AS, and most of the thing that you have here, I think you have already learned it in IGCSE. Okay. Okay, so if we go back through history, the discovery of the cell and its structure will not be possible with the help of Robert Hooke. Okay, I think you have learned this before, that Robert Hooke is a scientist who first discovered the structure of the cell. Okay, what did he do back then? I think it was somewhere in the 17th century, where he decided to examine a sample of plant material, and what he did was he chose a cork, right, a plant cork as one of his examples, put it down the microscope, and he was surprised to see a regular pattern, okay, on the structure. Okay, and the structure that he saw is actually the one that we have here, right? And Hooke detailed his observations of this tiny and previously unseen world in his book known as Micrographia. So this is the book that he wrote. Okay? And to him, the cork looked as if it was made of tiny pores, which he came to call cells, because they reminded him of the cells in a monastery. When Hooke first discovered the regular pattern that he later called as cells, he never thought that it would be the basic structure of living organisms, because the plant cork that he observed through his microscopes are dead. They are dead cells. However, 200 years later, somewhere around the 19th century, Matthias Jacob Schleiden and Theodor Schwann conducted further studies on plant and animal tissues, and they were the one who suggested that all plants are made of cells, right? And all animals are also made of basic unit known as cells, right? Which is what we have came to know by now. When you talk about the study of cell biology or cytology, two fundamentally different types of device or apparatus that can be used are the light microscope and the electron microscope. Both microscopes use a form of radiation in order to create a magnified image of the specimen being examined. Later on, we will look at this in detail. So for light microscope, the source of radiation used is visible light, while for electron microscope, the source of radiation used is the electrons. Okay, so we have discussed just now that the first microscope was invented and built by Robert Hooke in the mid of 17th century, where he discovered the first image of cells. But the actual golden age of light microscopy is around 19th century, right? Because in 200 years, the quality of the lenses that is used in microscope, improve. Right? They really improve a lot on the lenses. So this causes the scientists to become, keen in preparing materials so that they can observe it under the microscope. And the field where the scientists study the cell structure are known as cytology. The diagram that we have here is a diagram that shows the inner part of a light microscope and how it works. You will have a weekly hands-on session with the light microscope because one of the questions in P3 is about the handling and the usage of the microscope. Okay, the working principle is also shown here by the diagram, and it is basically quite straightforward. Yeah, you have the bulb here as the light source, so this will produce the light. Okay, and then the second part that you have here is the condenser iris diaphragm. Yes. So normally we will just call this as diaphragm. Okay, and the function of diaphragm is to create a narrow beam of light, and at the same time it controls how much light should be passed on to the lens. Right, so in a way this controls the brightness, okay, of the specimen. And then we have here what we call as the condenser lens, and the function of condenser lens is to focus the light beam exactly onto the specimen. Right? And then you have here the objective lens. So whatever light that has passed through the specimens will be picked up by the objective lens, and the objective lens will magnify the image of the specimen, right? And most light microscope, they have multiple objective lens with different magnifying powers, right? In the bio lab, okay, all of the microscope are equipped with 3 different objective lens. Right? The smallest magnification is 4 times. Then we have 10 times magnification. Okay, and the highest one would be 40 times magnification. Okay, and then the light beam will move in the eyepiece tube. So this is what we call as eyepiece tube before the light beam is focused on to the eyepiece lens to be observed. Right? So it's a very straightforward, simple working principle. With the improvement of the lenses and the sophisticated design of the modern microscope, a number of structures of the cells had been discovered. Some of the structures that are observable under the light microscope are shown here in figure 1.4, such as the mitochondria, the centriole, the Golgi body or the Golgi apparatus, nucleus, nucleolus, chromatin or chromosome, nuclear membrane, cell membrane, and perhaps a few other structures that might not be shown here. And with the help of the microscope as well, scientists have managed to discover that there are a few differences in terms of structure in plant cell and animal cells. Right? I think you have learned this, way back, from grade 7, and you're going to learn this again in A levels. But one of the most distinct features or differences between plant cell and animal cell is the presence of cell wall in plant cell, the absence of centriole in plant cell, the presence of vacuole in plant cell, the presence of chloroplast in plant cells, Okay, and also you can see that there is a structure known as plasmodesmata, okay, between two plant cells and etc. Right? So this discovery can only be done, okay, with the improvement of the microscope and the lenses. As you can see, figure 1.6 is a diagram of cells from the lining of the human cheek as seen through a light microscope, right? With some chemical staining, okay, you can really observe the structure of the nucleus, okay, and also the cytoplasm. And this is an actual diagram of a plant cell. Okay, this is a photomicrograph of a cells in a moss leaf, okay, magnified 400 times using a very high quality of light microscope, right? So you can see that, you can see the presence of this round green structure, which is actually the chloroplast. Okay. Besides the differences in terms of structure that we have discussed just now, there are a lot of common features between animal and plant cell as well, right? And in this chapter, we are going to to learn about those structures and those organelle in detail. Okay. And if you look at the picture that we have here, the plant cell and animal cell, you can clearly see that there are a lot of common organelles shared by the two, right? Common organelle and common structures, right? Common organelles such as mitochondria, nucleus, the endoplasmic reticulum, the Golgi apparatus, ribosomes, lysosome, okay, and etc. Common structures like the plasma membrane and also the nuclear membrane, right? And all of these organelles and structures will be discussed in further detail, okay, later on in this chapter. So this is the list of some common features of animal and plant cells that I was talking just now. And many of the cell contents are actually colorless and transparent. If you want to see them under the light microscope, they need to be stained with certain chemicals, right? And each cell has a nucleus, which is a relatively large structure that stains intensely and is therefore very conspicuous. The deeply staining material in the nucleus is called chromatin or chromosome. Right? So whenever you stain the nucleus, the content inside the nucleus will be stained as well, and the content of the nucleus is known as chromatin or chromosome, which is actually the DNA of the cell. And this is a list of differences between plant and animal cells that I've mentioned just now. You have centriole in animal, you have cell wall in plant, okay? And in terms of vacuole, uh, there is a very large central vacuole in plants, but you don't really have vacuole in animal cells. Even if you have it, the size is very small. And then chloroplasts can only be seen in plant and not in animal. Okay, with a light microscope, individual plant cells are more easily seen than animal cells because they are usually larger, and unlike animal cells, they are surrounded by the cell wall, right? So that makes it more obvious, okay, to be observed under the light microscope, okay, rather than just having the cell membrane alone. And the cell wall is relatively rigid because it contains fibers of cellulose, right? Which is a type of polysaccharide or carbohydrate which strengthen the wall. And the cell wall gives the plant cell, a definite shape. It prevents them from bursting when water enters the cell by osmosis. So it's quite opposite to what you have in animal cell. Plant cells are linked to their neighboring cells by means of fine strand of cytoplasm. So it's an extension of cytoplasm from one plant cell to another, known as plasmodesmata. So you can scroll back the video and you can check, okay, how plasmodesmata is organized in the plant cell. In cell studies, because it involves the measurement of a very fine and small structures inside the cell, so you need to use an appropriate measurement with appropriate unit, right? And in most of the time, the appropriate unit that is commonly used would be the millimeter, micrometer, and nanometer, right? In most of the past year papers, students will always be asked to convert, certain units to, okay, those units that we have here, right? Especially, micrometer, right? So to help you to memorize this, okay, always remember the conversion that I have down here, right? Always remember that 1 centimeter is equal to 10 millimeter, and 1 millimeter is equal to 1000 micrometer, and 1 micrometer is equal to 1000 nanometer, right? This will help you a lot because a lot of the question revolve around, converting certain units to certain units, right? Okay, next we will discuss on the size of biological structures. Okay, so when you talk about cell, how small can the cell get? Right? If you talk about bacterial cell, okay, the smallest size that a bacteria can have is somewhere around 400 nanometers, right? And if you talk about a normal plant and animal cell, okay, the smallest plant and animal cell can be as small as 1 micrometer or 1000 nanometers, right? So that's, a general idea on how small the world of cell is. However, there is a limitation on how small of an object that the human eye can see, right? Because the human eye can only see structures or objects, uh, as small as 50 micrometers. Any objects or structures smaller than that, so we could not actually see it with our naked eyes, right? So smallest structures such as algal cell, the thickness of leaf, or paramecium are still objects that can be seen by the eyes without any visual aid, such as microscope. Okay. And if you still want to observe structures or objects that are smaller than 50 micrometers, then the only way to do that is by observing them under the light microscope. Because the light microscope enables you to see structures or objects as small as 0.2 micrometers, or roughly about 200 nanometers, right? So if you want to look at the diameter of a plant cell or animal cell, if you want to look at the width of human hair, and if you want to look at the diameter of mitochondrion or diameter of bacterium better, so use light microscope, because it will magnify those objects, okay, to a size that is observable to the eyes. So what if you want to observe a structure or an object which is much smaller than 0.2 micrometer, okay, which is the size that can be observed using the light microscope. So what if you want to observe a structure or an object that is much smaller than that? Okay, because in reality, a lot of structures inside the cell, okay, will be much smaller than 0.2 micrometer. Okay? For example, if you talk about a ribosome, right, the size of ribosome is roughly about 25 nanometers, right? 25 nanometers. Okay, so using light microscope, okay, you will not be able to observe the structures of chromosome. Okay, so the only way, okay, to observe, structures that are smaller than 0.2 micrometer is by using the electron microscope. Right? Because electron microscope will enable you to observe structures as small as 0.5 nanometers. Okay? So with the help of the electron microscope, you can observe a lot of very small fine structures inside the cell, such as the ribosome, okay, the thickness of the plasma membrane, and also the diameter of DNA. Okay, in this chapter, you are also going to learn how to calculate magnification, right? Because in most of the past years, magnification is one of the questions that will always come out every single year. So what is magnification? Magnification is the number of times larger an image is compared with the real size of the object. I think you might be familiar with the formula of the magnification. I think you might have already learned this in IGCSE, but whatever it is, okay, you will still have this in A levels. So magnification is given as size of image over the actual size of specimen. Okay, so let's look at some examples of questions on magnification. Right? So let's look at the first one. It is given to you that the real length of the diagram is 80 micrometer. Measured length is 60 millimeter. And you are asked to find out the magnification of the drawing. And I think this is quite easy. Okay, you need to go back to the formula. Formula of the magnification is the size of image divided by the actual size of the specimen, right? And the size of image here is 60 mm, and the actual size of the image is 80 micrometer, right? So you need to standardize the unit first, right? If it was me, I would change the millimeter to micrometer and then plug in all the figures into the formula. So 60 millimeter is equal to 60,000 micrometer, right? Because I've given you the list of conversion just now. 1 millimeter is equal to 1,000 micrometer. Remember? So 60 millimeter is equal to 60,000 micrometer. So therefore, to solve this question, 60,000 micrometer divided by 80 micrometers, and you will get the answer as 750 times, right? So the magnification is 750 times. Okay, so the next example, okay, you are given a scale bar, okay, and the actual size of the scale bar is given there as 6 micrometers, right? And when you measure it using a ruler, okay, it turns out that the size of the scale bar is 36 millimeters, okay? And you are asked to find out what is the magnification. So this is the same as the previous one. Formula of magnification is the size of image divided by the actual size of the specimen. And in this case, the size of image is 36 millimeter, and the actual size of the image is 6 micrometers. So convert 36 millimeters to micrometer first. So 36 millimeters is the same as 36,000 micrometers. Okay, and once you have that, plug it into the formula, and you will get 36,000 micrometers divided by 6 micrometers. So the answer will be, the magnification is 6,000 times, right? 6,000 times. Okay, and the third example is slightly different from the first two, where you are now given magnification, right? You are now given the actual magnification, and you are also given the size of image, okay, shown here as the measured length, which is 40 millimeters. Okay, and you are asked to find out what are the actual size, okay, of the specimen. Right, so you need to play around with the formula, and to find out what the actual size is. So the formula will now be magnification over the measured length, right? So once you divide magnification with the measured length, okay, you will get roughly about 0.0071 millimeters. Okay, but normally the questions will ask you to provide your answer in micrometer, right? So 0.0071 millimeter, okay, is the same as 7.1 micrometer, or you can just write it down as 7 micrometers. Okay. Besides magnification, another term that you need to know when you use a microscope is resolution. And resolution is defined as the ability to distinguish between two separate points. Okay, and the maximum resolution of light microscope is 200 nanometers, while the electron microscope has higher resolution, which is 0.5 nanometers. Okay, let's say you are observing two dots using a light microscope, and the distance between the two dots is 50 nanometers apart. So observing both of the dots using a light microscope will cause you to see them as one dot. Why? Because the resolution of light microscope is only down to 200 nanometers. Any object or gap smaller than that, okay, the light microscope will not be able to differentiate it. Okay, so in order to see the two dots clearly, you can use an electron microscope because it has higher resolution that can differentiate objects or gaps as small as 0.5 nanometers. So that is why when you look at these two diagrams, you can tell that the one on the left is most probably observed with a light microscope, while the one on your right is most probably observed with an electron microscope. Because microscope with higher resolution power will allow you to see all the details in the specimen, as it can clearly differentiate all the small objects, gaps, groove, and any structure as small as 0.5 nanometers, which a light microscope can't. It is possible to take photograph, okay, on the left, right, and magnify it as much as we want. Okay, so you can make it as big as you want, but we will still not going to see much of detail in the magnified picture. In other words, magnifying the diagram will not improve its resolution. Remember when I said earlier that both electron and light microscope use radiation to observe the specimens. The radiation that is used comes from a range of different wavelengths in the electromagnetic spectrum. And as you can see in theory, there is no limit on how short or how long a wavelength can be. The higher the energy that is given, the shorter the wavelength is going to turn up. However, in light microscope, the radiation that is used is from those of the visible wavelength spectrum, which is between 400 nanometers to 700 nanometers. And that is why with the radiation emitted by the visible wavelength, you can only see object as small as 200 nanometers, because the general rule of resolution is that the limit of resolution is about half of the wavelength of the radiation used to view the specimen. So if you use the visible light wavelength to observe the specimen, so the smallest object that you can see is half of the smallest wavelength in the visible light spectrum, which is 200 nanometers. Okay, so hope that clear things up. For example, why can we see nucleus with the light microscope and not ribosome? Okay, so there are a lot of organelles inside the cell. Okay, so let's just choose two, the nucleus and ribosome. Why can you see nucleus with the light microscope, but you can't see any ribosome using the same light microscope? This is because the size of nucleus is about 6,000 nanometers in average. And the shortest wavelength of the visible light spectrum is 400 nanometers, right? So the size of the nucleus is large enough to interfere with the wavelength of the visible light, causing it to be reflected back, okay, to our eyes to be seen. Right? So the object is large enough that causes the wavelength to be interfered, and some of it will be reflected back to our eyes so that we can observe it, right? So that's the theory. Okay, and the diagram that we have here shows how a mitochondrion, okay, with the size of 1000 nanometers, okay, is able to interfere with the wavelength of the visible light. And like I said just now, some of the wavelength will be reflected back, okay, for us to see. Okay, always remember about the general rule of resolution, which says that the limit of resolution is about half of the wavelength used to view the specimen. So using a light microscope, okay, the smallest object that you can see is 200 nanometers, right? Because that is the half of the size of the visible light wavelength, because the shortest wavelength of the visible light is 400 nanometers. So half of that is 200 nanometers. So that will be the resolution of the visible light microscope, right? Smallest object that you can see would be 200 nanometers, which is the resolution. So how about ribosome? The size of an average ribosome is 22 nanometers, which is way smaller than the wavelength of the visible light, right? So can ribosome interfere with the wavelength of the visible light and reflected back for us to see? So I would like you guys to think about this during the weekend, because the answer lies in the next part of the chapter when we discuss about the electron microscope. So I will stop here for today. Okay, inshallah, we are going to continue with the rest of the chapter next week. Okay, so please check the assignment on the Google Classroom and try to complete it and submit it back, okay, through the Google Classroom as well. Okay, guys, Assalamualaikum warahmatullahi wabarakatuh.